Welcome back, everyone. Get ready for another deep dive, and this time, we're taking on a topic you're already an expert in taste.
Well, I'm excited to see what you found. I always love learning more, especially about something so fundamental to our everyday lives.
Exactly. We experience it every day, but I bet most of us haven't stopped to think about the amazing biology behind it. How does our tongue actually translate chemicals into those sensations of sweet, sour, salty, bitter, and umami? You sent over some research that really dives into this, and I was especially intrigued by these things called GPCRs and ion channels.
Ah, yes. Those are the real stars of the show when it comes to taste. It's just incredible how these tiny molecules work together to give us this amazing sensory experience we call taste.
Okay, so let's break it down. You sent me a ton of fascinating stuff, papers on thyroid hormone receptors, research on glycine receptors in the spinal cord, even some deep dives into cyclic AMP signaling. Wow, seems like a pretty diverse mix. How does all of that connect back to taste receptors?
Well, you're right. It might seem like a bit of a detour at first glance, but all of those sources actually provide a broader context for understanding how cells communicate and respond to different signals. And that foundation is going to help us really appreciate the intricate mechanisms that are at play in taste perception.
Okay, so it's like setting the stage before the main act. I like it. So if we want to understand how we actually taste, where should we start?
Well, let's begin with the taste buds themselves. They're like tiny sensory outposts scattered all over your tongue, each containing a mix of 50 to 100 specialized taste receptor cells.
So each taste bud is like a little city of flavor detectors, huh? That's pretty incredible.
Yeah.
Are all of those receptor cells the same, though? Or
Not at all. And that's where those GPCRs and ion channels you mentioned come in. They're like specialized sensors within those taste bud cities. And each one is tuned to detect a specific type of taste molecule.
so some cells are wired for sweet, some for sour, and so on. That's pretty cool.
Exactly. Sweet, bitter, and umami tastes, they all depend on those GPCRs, G protein-coupled receptors. They act like highly selective locks, each waiting for a specific taste molecule to come along and unlock it.
Okay, I think I'm starting to see how this works. So, for example, a sugar molecule would be the key to unlock the sweet receptor.
You got it. And when that sweet key fits into the sweet lock, it triggers a cascade of signals within the taste receptor cell. Think of it like setting off a chain reaction, which ultimately sends a message to your brain saying, hey, this is sweet.
And then our brain interprets that signal as the sensation of sweetness, right?
Mm.
Wow. It's incredible how our bodies translate these tiny molecular interactions into something that we experience as taste.
It really is remarkable. Now, for bitter and umami tastes, it's a similar story, but with different types of GPCRs, each tuned to detect a specific set of molecules. For instance, certain amino acids, like glutamate, are the keys that unlock those umami receptors.
Right, glutamate. That's what gives us that savory deliciousness we find in foods like Parmesan cheese or mushrooms. I'm really liking this whole lock and key analogy. It definitely makes understanding these complex molecular interactions so much easier. So what about sour and salty? Do they use GPCRs too?
Those are a bit different. Instead of relying on those intricate lock and key mechanisms, sour and salty utilize ion channels.
Ion channels, right. Those are the protein tunnels that allow charged particles to flow in and out of cells.
That's right. Think of them like tiny gates guarding the entrance to the taste receptor cell. For sour taste, the gatekeeper is particularly sensitive to hydrogen ions, those tiny charged particles that make things acidic.
So the more acidic something is, the more those sour gates open up, sending a stronger signal to the brain.
Exactly. It's a direct link between the chemical environment and the electrical signal. And then for salty tastes, those gates respond mainly to sodium ions, which of course is the main component of table salt.
Uh-huh. So when you sprinkle some salt on your food, you're essentially flooding your taste buds with those sodium ions, right? And that's what triggers those salty gates to open wide. Makes sense. This is making me think about all those old school tongue maps we saw back in elementary school. The ones that claim different parts of your tongue were responsible for different taste. Is that actually true?
It's funny how that myth persists, but in reality, while you do have clusters of taste buds in different areas of your tongue, they're pretty much all over. And each taste bud can detect all five basic tastes: sweet, sour, salty, bitter, and umami.
So those tongue maps are officially busted. I am definitely going to share that little nugget of knowledge at my next dinner party.
It's always fun to debunk those persistent myths. And here's another interesting fact to add to your arsenal. You even have taste receptors in your gut.
Wait, what? In our stomachs.
Yep. They don't contribute to your conscious experience of taste, but they play a pretty crucial role in things like digestion and nutrient sensing.
Wow, our bodies are so intricate. But let's get back to the tongue for a second. If each taste bud can detect all 5 tastes, how does our brain actually figure out what we're tasting? Is it just about which receptor cells are activated?
It's actually much more nuanced than that. It's not just a simple one receptor, one taste system. Your brain relies on a complex code, a symphony of signals, to really decipher the flavors you're experiencing.
So it's like the brain is reading a complex language of taste, with different combinations of signals creating different flavor perceptions.
That's a great way to put it. The brain takes into account which receptors are activated, the intensity of those signals, and even where on the tongue those signals are coming from.
So it's not just about the individual notes, but about the entire composition, the way those notes blend together to create a unique flavor experience.
Exactly. And this complexity is what allows us to distinguish between those subtle variations within a single taste category. For example, you can tell the difference between the sweetness of honey and the sweetness of an artificial sweetener, even though they both technically activate the same type of sweet receptor.
That is amazing. Our brains are like little flavor conductors, taking all these signals from our taste buds and creating a whole symphony of taste sensations. This is making me think about all the possibilities for creating new and interesting flavors.
You're hitting on a really exciting area. Understanding these molecular mechanisms is actually revolutionizing the food industry. Scientists are using this knowledge to design foods and beverages that target specific taste receptors, creating entirely new flavor experiences.
That sounds straight out of science fiction. So instead of just relying on traditional ingredients, now we can actually create flavors by manipulating those taste receptor pathways.
Precisely. It's like having a toolbox of molecular building blocks for flavor. For instance, researchers are developing low-calorie sweeteners that activate those sweet receptors without the negative health effects of sugar. Imagine enjoying all the sweetness of your favorite desserts, but without the guilt.
That would be a game changer for sure. But are there other applications for this kind of research beyond just sweeteners?
Absolutely. Think about medications. Many life-saving drugs have a notoriously bitter taste, making them difficult to swallow, especially for children. But by understanding how those bitter receptors work, scientists are finding ways to mask that bitterness, making those medications much more palatable and easier to take.
That's incredible. It sounds like this knowledge about taste receptors isn't just about creating delicious food, it's about improving people's health as well.
Exactly. And it even has implications for cultural understanding. By studying how different populations perceive taste, we can actually create food and beverages that appeal to specific cultural preferences. Imagine bridging cultural divides through the language of flavor.
This is truly mind-blowing. I can't wait to dig even deeper into this. But before we do, let's take a closer look at those GPCRs involved in detecting sweet, bitter, and umami tastes. You mentioned these things called T1R and T2R receptors. What are they all about?
They're the molecular workhorses of those taste modalities. T1R receptors are particularly fascinating because they work in pairs, kind of like a dynamic duo. For example, the pairing of T1R1 and T1R3 creates a receptor that's specifically tuned to detect umami. And for sweet taste, it's the T1R2 and T1R3 pairing that forms the sweet taste receptor.
So it's like a mix and match system with different combinations of T1R receptors creating detectors for different tastes.
Exactly. Now, the T2R receptors, on the other hand, are soloists. Each one is dedicated to detecting a specific type of bitter compound.
Okay, so they each have their own specialty. Interestingly, the research you sent mentioned that humans actually have many more types of bitter receptors than sweet receptors. Why is that? Is it because bitter things are generally unpleasant, so we need more ways to detect them and avoid them?
That's a great question. It actually reflects the evolutionary importance of being able to detect potentially harmful substances in our environment. You see, many toxins found in plants and other natural sources are bitter. Having a wide array of bitter receptors allows us to quickly identify and avoid ingesting things that could be dangerous.
So it's like our bitter taste receptors are our internal. Now, we've talked about how these GPCRs work like locks and keys, with specific molecules acting as keys to unlock specific taste sensations. Can you give us some examples of what those keys are for the different taste locks?
Sure. For sweet taste, the keys are sugars like glucose and fructose, the kind you find naturally in fruits and honey. But it's not just about natural sugars. Scientists have designed artificial sweeteners that actually mimic the shape and interaction of sugars with that T1R to T1R3 receptor.
So they can trigger that same sweet sensation without the calories.
Precisely. Now, for umami, the key players are certain amino acids, particularly glutamate. Think about savory flavors like those found in meat, cheese, and mushrooms. They all contain glutamate, which triggers those umami receptors and gives us that satisfying, savory taste.
It's like a gourmet lock and key system, where each key unlocks a specific delicious sensation.
I like that. And for bitter taste, well, the keys are much more diverse. This reflects the wide range of bitter compounds found in nature. These can include alkaloids, phenolic compounds, and certain salts, all of which can activate different combinations of those T2R receptors.
This is making me realize that our sense of taste is far more complex than I ever imagined. It's not just about 5 basic tastes, it's about a whole symphony of signals from a multitude of receptors, all orchestrated by our brains to create this rich tapestry of flavor perception. It's truly remarkable.
It is remarkable, and we've only just begun to explore the depths of this intricate system. It really is remarkable. And honestly, we've only just begun to explore the depths of this intricate system.
Well, I'm ready to go deeper. You mentioned earlier that some of the research I sent over about other types of receptors actually helps set the stage for understanding taste. There was one in particular that I found really intriguing, the one about cyclic AMP signaling and these things called microdomains. It sounded pretty complex. Can you break it down for me and for our listeners and maybe explain how it relates to taste?
Absolutely. You see, for a long time, scientists thought of that cyclic AMP signaling as sort of a global announcement system within a cell. So when a GPCR was activated, it would trigger the production of cyclic AMP, which would then spread throughout the entire cell, kind of like a loudspeaker broadcasting a message.
So any part of the cell could hear that message and respond accordingly.
Exactly. That was kind of the prevailing theory for a while. But then researchers started to notice some puzzling discrepancies. They found that different GPCRs, even though they both activated the same cyclic AMP pathway, could actually produce very different effects on the cell.
So 2 different signals using the same pathway, but ending up with different outcomes. Huh, that doesn't quite fit with the loudspeaker analogy, does it?
Exactly. It's like sending the same message over the loudspeaker, but getting 2 totally different responses from different parts of the room. So that's where this concept of microdomains comes in. It suggests that cyclic AMP signaling isn't this global broadcast, but rather it's a series of more targeted messages delivered to very specific regions within the cell.
So instead of one loudspeaker, we've got multiple loudspeakers, each broadcasting to a specific section of the room.
Precisely, and this helps explain how different GPCRs, even though they're activating the same pathway, can produce those distinct effects. They might be activating cyclic AMP production in different microdomains, which leads to the activation of different downstream targets. It's like having a highly organized signaling network within the cell where those messages are delivered with incredible precision and specificity.
Okay, that makes sense. So how does this idea of microdomains apply to our taste receptors then?
Well, remember that those taste receptor cells use those GPCRs to detect sweet, bitter, and umami tastes. So it's definitely possible that microdomains play a role in organizing these signaling pathways within those taste cells, making sure that the right signals get to the right places to generate those very distinct taste sensations.
So in essence, each taste, sweet, bitter, umami, might have its own little designated microdomain within the taste cell, its own little section of the room with its own dedicated loudspeaker.
That's a very plausible hypothesis. It would definitely add another layer of complexity and specificity to our understanding of taste perception. And while the research on these cyclic AMP microdomains isn't directly focused on taste receptors, it does give us a helpful framework for thinking about how these signaling pathways might be organized within those taste bud cities.
This is fascinating. It's like we're discovering this whole hidden level of organization within our taste buds, a whole network of specialized communication channels.
It really highlights the incredible complexity of even seemingly simplest of biological processes. And speaking of complexity, let's talk about some of the key players in these microdomains. You also sent me some research about these things called kinase anchoring proteins, or AKPs for short.
Yes, those AKPs sounded like they play a pretty important role in this whole microdomain idea. What exactly do they do?
AKPs are like the architects of these microdomains. Imagine them as this molecular scaffolding, bringing together various components of the signaling pathways and organizing them into these distinct signaling hubs. They basically ensure that all the right players are in the right place at the right time.
So they're like the event planners for those targeted messages, making sure everything runs smoothly.
Exactly. And in the context of that cyclic AMP signaling, they help prevent those signals from becoming too global and ensure that that cyclic AMP message is delivered specifically to its intended audience within the cell.
It's incredible how these tiny molecules can orchestrate such complex signaling events within our cells, especially within our taste buds.
Absolutely. It really highlights the importance of that spatial organization in cellular signaling. It's not just about which molecules are present, it's about where they're located and how they interact with each other within these very specific microdomains.
This whole concept of microdomains in these AKPs is pretty mind-blowing. It's like each receptor cell has its own internal postal system, with those AKPs acting as the mail carriers, delivering those taste messages to the right addresses.
That's a great analogy. And just like a postal system needs different types of mail carriers for different types of deliveries, there are different types of AKPs, each with their own unique set of binding partners and functions.
Okay, so let's talk about some of those specific AKPs. One of the sources that you sent me mentioned something called an amphipathic helix. It seemed to be involved in how AKPs bind to those protein kinase A molecules. Can you explain what an amphipathic helix is and why it's important?
Certainly. Imagine a spiral staircase. Now imagine that one side of that staircase absolutely loves water. We call that hydrophilic, while the other side repels water, hydrophobic. That's essentially what an amphipathic helix is, a twisted structure with this dual nature.
So it's like a staircase where one side is painted with water-resistant paint and the other side is left bare.
That's a great way to visualize it. Now, this amphipathic nature is absolutely crucial for how those AKKPs interact with that protein kinase A, or PKA. You remember that PKA is a key enzyme involved in those signaling pathways. So the hydrophobic side of the helix, the water-repelling side, nestles into a specific groove on PKA, while the hydrophilic side interacts with the surrounding watery environment of the cell.
So it's like one side snuggles in for a close embrace, while the other side makes sure that everything stays stable in that cellular environment.
Precisely. This dual nature allows AKKs to anchor PKA with remarkable specificity, which creates those microdomains of cyclic AMP signaling. It's a beautiful example of how protein structure dictates function at a molecular level.
It's fascinating how such a subtle detail, this dual nature of the helix, can have such a huge impact on how these signaling pathways are actually organized.
Absolutely. And it highlights just how much precision and elegance exists within our cells. Speaking of PKA, did you know that there are actually different types of this enzyme? Specifically, there's type 1 and type 2 PKA, and they don't interact with AKPs in quite the same way.
Well, that's interesting. So what's the difference between those two types of PKA, and why does it matter which one those AKPs bind to?
Well, type 1 and type 2 PKA are kind of like siblings with slightly different personalities. They both have the same basic job to phosphorylate other proteins and modify their activity, but they differ in their regulatory subunits, the parts of the enzyme that control their activity.
So it's like they have different on and off switches.
Exactly. Now, type 1 PKA typically likes to roam freely throughout the cell's cytoplasm, but type 2 PKA prefers to be anchored to specific locations thanks to its affinity for those AKAs.
So type 2 PKA is the one that's more likely to be involved in those microdomains, those organized signaling hubs, because it can be tethered in place by those AKPs.
Precisely. And this anchoring is crucial for actually creating and maintaining those localized signaling compartments, ensuring that those right signals get to the right places.
It's amazing how these subtle differences in their personalities can have such a big impact on their roles within the cell.
And those differences all come down to the specific structures of their regulatory subunits. The regulatory subunits of type 2 PKA actually have a special binding motif, a small region that perfectly matches the shape of that amphipathic helix on those AKPs, allowing them to connect like two puzzle pieces.
So it's like type 2 PKA has a special hook that latches onto that AKP, ensuring that it stays put in those microdomains.
That's a great way to put it. Now, the source you sent me also goes into detail about the structure of these regulatory subunits. Specifically, it mentions these things called dimerization and docking domains. Have you ever heard of those?
Vaguely. I could definitely use a refresher, though. Can you break those down for us and maybe explain how they relate to all of this?
Of course. Remember how we were talking about those regulatory subunits acting like gatekeepers for PKA? Well, those gatekeepers don't actually work alone. They pair up, forming a dynamic duo called a dimer.
So instead of one guard at the gate, we have two working together.
Exactly. And the region responsible for this pairing, for those two regulatory subunits coming together, is aptly named the dimerization domain.
It's like a handshake between those two guards, forming a strong bond.
A perfect analogy. Now, in addition to this dimerization domain, each regulatory subunit also has this special region called the docking domain.
Okay, and that's the part that interacts with that AKAP, like a
Yeah.
speckled badge that allows access to those microdomains.
You got it. That docking domain contains a specific binding motif, a molecular handshake, if you will, that interacts perfectly with that amphipathic helix of those AKPs, forming this really stable connection.
So it's this intricate interplay between that dimerization, docking, and the amphipathic helix that allows AKAPs to organize that pKa and create those spatially defined signaling compartments.
Precisely. It's like a beautifully orchestrated molecular dance, and it's all happening within the tiny confines of a cell.
This level of detail is incredible. I never realized just how much was going on beneath the surface of something as simple as tasting. I am still a little fuzzy on this whole helix-turn-helix motif that the source mentioned, though. What is that, and why is it so important?
Yes, the helix-turn-helix motif. It's a very common structural element found in a lot of proteins, and it plays a really crucial role in this AK-pKa interaction. Imagine a protein chain folding upon itself to form a helix, kind of like that spiral staircase we were talking about earlier. Then it makes a sharp turn and forms another helix, creating this helix-turn-helix structure.
So it's like two spiral staircases connected by a landing.
Precisely.
Yeah.
Now, in the case of the pKa regulatory subunit, this helix-turn-helix motif is located within that docking domain. And guess what? It's actually the hydrophobic face of this motif, that water-repelling side, that interacts with the amphipathic helix of the AKKPs, anchoring the pKa in place.
So it's like the helix-turn-helix motif provides this perfectly shaped groove for that amphipathic helix to nestle into, like two puzzle pieces fitting together.
You're getting it. It's this precise structural fit that allows AKPs to bind with such incredible specificity, making sure that the pKa is directed to the right location within the cell to create those organized signaling hubs.
This is making me realize just how intricate and elegant the molecular world really is. Every detail, every shape, every interaction is playing a crucial role in orchestrating these really complex cellular processes. It's like watching a molecular ballet with each molecule moving and interacting with such precision and purpose.
And that ballet is happening within your taste buds every time you savor a flavor.
Now, the source also mentioned that the dimerization and docking domain of type 2 pKa is a bit different from that of type 1 pKa. Can you explain those differences and why they matter in terms of that AKP binding?
Certainly. The key difference actually lies in an extra alpha helix that's present in the dimerization and docking domain of type 1 pKa. So it's like type 1 pKa is wearing this bulky coat that prevents it from getting a good grip on that AKP.
So it's got like a type 1 pKa has this extra appendage that just gets in the way, making it harder for those AKPs to latch on.
That's a great way to visualize it. And that's exactly why type 1 pKa prefers to roam freely in that cytoplasm, while type 2 pKa, with its more streamlined dimerization and docker domain, is more readily anchored by those AKPs.
It's amazing how such a small structural difference can have such a huge impact on how these proteins interact, and ultimately on their functions within the cell.
It really highlights the importance of precision and specificity in the molecular world. It makes you wonder about the potential implications for taste perception. Could different types of pKa be involved in generating different taste sensations? Could that balance between type 1 and type 2 pKa actually influence how we perceive those flavors?
Those are fantastic questions. It seems like we've just scratched the surface of understanding these molecular intricacies of taste.
Absolutely. And the more we delve into these details, the more we appreciate the complexity and the elegance of this sensory system.
Now, we've talked a lot about how these taste receptors are organized within these microdomains and how they interact with various signaling molecules. But I'm curious about the final step in this whole process. How do those receptors actually generate a signal that our brains can interpret? It's like we've been focusing on the ingredients and the cooking process, but we haven't actually tasted the final dish.
That's a great point. You're talking about signal transduction. That's the process of converting a stimulus, like a taste molecule binding to a receptor, into an electrical signal that can be transmitted to the brain. It's like translating the language of chemistry into the language of our nervous system.
So it's like taking those carefully prepared ingredients and transforming them into a delicious, brain-ready meal.
Exactly. And this transformation involves a whole series of molecular events, like a cascade of signaling pathways that ultimately lead to the generation of an electrical impulse, the language that your brain actually understands.
Okay, walk us through that cascade. How does a taste molecule binding to a receptor on the tongue eventually lead to a signal that our brain can understand?
Let's revisit those GPCRs, the receptors for sweet, bitter, and umami. Remember that lock and key analogy? So when a taste molecule binds to its corresponding GPCR, it's like turning a key in a lock. It activates the receptor.
Right, and that sets off that chain reaction inside the taste receptor cell.
Precisely. That activated GPCR interacts with a protein called a G protein, which is located just beneath the cell membrane. So it's like turning that key in the lock triggers this lever on the inside.
I'm starting to see the chain of events here. So what does that G protein do?
The G protein acts as a molecular messenger. It relays the signal from that activated GPCR to other proteins within the cell. Specifically, it activates an enzyme called adenylate cyclase.
And adenylate cyclase is responsible for producing cyclic AMP, right? We
Right.
talked about that earlier.
Or on fire. Cyclic AMP, or CAMP, acts as what we call a second messenger, carrying the message forward, like a relay runner passing the baton.
Okay, so what's the next step in the relay?
Cyclic AMP activates another enzyme called protein kinase A, or PKA, which you might recall is the same enzyme that we discussed earlier in relation to those AKPs and microdomains.
Right, PKA is the one that phosphorylates other proteins, changing their activity. So it's like flipping a switch, triggering a cascade of events.
That's a great way to think about it. In the context of taste, this PKA activation ultimately causes ion channels within the taste cell to either open or close.
Ah, those ion channels again. The gates that control the flow of charged particles across the cell membrane. So are we finally at the point where the electrical signal is generated?
We're getting there. When those ion channels open or close, it changes the electrical charge across the taste cell's membrane. So this change in charge, this electrical surge, is detected by sensory neurons that are connected to the taste cell.
So those sensory neurons are like the wires that carry the electrical signal from the taste bud all the way to the brain.
You got it. And once that signal reaches the brain, it's interpreted as a very specific taste, sweet, bitter, or umami, depending on which GPCR was initially activated.
Wow. It's amazing how this complex cascade of molecular events, this relay race of signals, can be triggered by something that simple as a molecule binding to a receptor on our tongues.
It really highlights just how incredibly powerful and elegant biological signaling systems can be. You might be wondering how this process differs for those sour and salty tastes, which are detected by those ion channels rather than GPCRs, right?
That's exactly what I was just about to ask. Does it involve a similar cascade of events?
For sour and salty, the signaling pathway is actually a bit more direct. Remember, that sour taste is triggered by those tiny charged particles called hydrogen ions.
Right. The more acidic something is, the more hydrogen ions it has, the more sour it tastes. But how do those ions actually generate that signal?
Well, instead of binding to those GPCRs, hydrogen ions actually interact directly with specific ion channels on the taste cell's membrane, causing them to open. It's not really a lock and key mechanism, it's more like a direct push on a gate.
So when those gates open, what happens?
When those sour gates open up, it allows positively charged ions, primarily sodium ions, to rush into the taste cell. So this influx of positive charge changes the electrical charge across that membrane, generating the signal that's then sent to the brain.
So even though it's a different mechanism than the GPCR pathway, the end result is the same, right? An electrical signal that the brain interprets as that sour taste.
Exactly. It's a more straightforward process, but just as effective.
And what about that salty taste?
For salty taste, it's a very similar story. Those sodium ions from salt interact directly with specific ion channels, causing them to open and allowing more sodium ions to flow into that taste cell. So again, this influx of positive charge depolarizes the cell, generating that electrical signal that's then interpreted by the brain as salty.
It's incredible how our sense of taste has evolved to utilize both these direct ion channel interactions and those more complex GPCR pathways, all to create this incredibly rich tapestry of flavor perception.
It really speaks to the ingenuity of those biological systems. Now, we've explored how those individual taste receptor cells actually generate signals, but how does your brain take all of that information, all of those electrical impulses from your tongue, and actually create a cohesive perception of flavor? It's not just about individual tastes, is it? It's about the overall experience.
That's a great point. It's like we've been focusing on the individual instruments in an orchestra, but what about the conductor? How does the brain actually orchestrate this whole symphony of taste sensations?
That's where things get even more fascinating. Our perception of flavor is actually a multisensory experience. It's not just about taste, but about the integration of taste with other senses. Smell, texture, temperature, even visual cues play a role.
So it's like our brains are creating this multi-dimensional flavor map. They're combining information from all of our senses to create a more complete and nuanced experience.
Precisely. And this integration happens so seamlessly we don't even realize it's happening. Let's start with smell. Smell plays a huge role in how we perceive flavor. When you eat, odor molecules from the food travel up the back of your throat to your nasal cavity where they interact with those olfactory receptors. Those are the receptors responsible for your sense of smell.
So while our tongues are busy detecting those basic tastes, our noses are adding another layer of information, a whole bouquet of aromas.
Exactly. And these olfactory signals are then sent to the brain where they're integrated with the signals from our taste receptors. This combination of taste and smell is actually what gives us that really complex perception of flavor.
That makes so much sense. I've definitely noticed that when I have a cold and my nose is all stuffed up, everything tastes so bland and muted. It's like the flavor is missing a key ingredient.
That's because your sense of smell is compromised, so you're not getting that full flavor experience. Your brain isn't able to blend those taste and smell signals together as effectively.
It's incredible how much our sense of smell actually contributes to our enjoyment of food. You mentioned that other senses are involved too, though. What about texture? How does that influence flavor?
Texture plays a really crucial role as well. The way food feels in your mouth, whether it's crunchy, creamy, chewy, or smooth, sends signals to your brain that are also integrated with those taste and smell signals. This further enriches your flavor experience. Think about the difference between biting into a crisp apple and sipping applesauce. Even though they have the same basic apple flavor, that difference in texture creates a totally different sensory experience.
You're so right. I never thought about it that way, but the texture definitely changes how I perceive the flavor. What about temperature? Does that play a role too?
Temperature is definitely a factor. It can actually influence how volatile the aroma molecules in food are, which affects how strongly we smell them. For example, hot coffee typically have a much stronger aroma than cold coffee because the heat helps those aroma molecules escape into the air. This means they can more easily reach our olfactory receptors, intensifying that whole flavor experience.
So temperature can actually enhance or diminish certain flavors just by affecting how we perceive those aromas.
Exactly. And it's not just about smell. Temperature can actually directly affect those taste receptors as well. Think about how a cold beverage can kind of numb your taste buds a little bit, making the flavors less intense.
This is blowing my mind. I never realized how interconnected all of our senses are when it comes to experiencing flavor.
And there's one more sense that plays a subtle but important role: sight.
Really.
Okay.
What does what we see have to do with how something tastes?
Think about it this way. The appearance of food, its color, shape, and presentation, can actually set expectations that influence how we perceive its taste. Imagine being served a beautiful, vibrant salad versus a wilted brown one. Even if those ingredients are identical, that visual perception might lead you to experience those flavors differently.
That's so true. We eat with our eyes first, as they say.
Exactly. Our brains are constantly integrating information from all of our senses to create a really cohesive and multi-layered flavor experience. It's a testament to the incredible processing power of our brains.
This whole discussion has completely transformed my understanding of flavor. I never realized how many factors were at play, how many different signals our brains are juggling to create this symphony of sensations.
It's a truly remarkable system, and we're honestly only just beginning to unravel its intricacies. But even with what we know now, we can already appreciate the incredible interplay of biology, chemistry, and even psychology that goes into every bite we take.
I'm starting to think about food in a whole new way. It's like every meal is a sensory adventure. A masterpiece of biological orchestration.
Well said. And on that note, I'm suddenly feeling quite peckish.
Me too. I think it's time to put all of this newfound knowledge to the test and savor some delicious flavors, armed with this deeper understanding of all the incredible processes that are happening both on our tongues and in our brains.
I couldn't agree more. Let's indulge in a culinary symphony.
Okay, ready for round 3 of this taste bud adventure. We've explored the intricate world of taste receptors, those GPCRs and ion channels, and we even went on that fascinating journey into the realm of microdomains and AKPs. What other gems have you uncovered in those research materials I sent over?
Well, you included a couple of sources that, while not directly about taste, provide some interesting parallels and insights. One of them talked about those thyroid hormone receptors. They're not actually involved in taste perception, but they offer a glimpse into the broader world of receptor biology.
I remember that one. It talked about how those receptors can act as both gene silencers and gene activators,
Yeah.
depending on whether a thyroid hormone is bound to them. It's like they have these dual personalities.
Exactly. And that ability to switch between different modes of action is actually crucial for regulating a whole wide range of physiological processes, everything from metabolism to development. The source even highlighted how mutations in those receptors can actually lead to various diseases. This emphasizes their critical role in maintaining our health.
That's a great reminder that understanding how these receptors work, it can have far-reaching implications,
Yeah.
way beyond just taste perception.
Absolutely. And while taste receptors and thyroid hormone receptors have very different roles in the body, they actually share some fundamental mechanisms. Both rely on the binding of a specific molecule, what we call a ligand, to trigger those changes in their shape. Ultimately, this influences gene expression.
So by studying one type of receptor, we can actually gain insights into the general principles of how other receptors might function, even if they're regulating totally different processes.
Precisely. It's about recognizing those common threads that connect those seemingly disparate biological systems.
That's a great point. Now, there was another source that initially left me scratching my head a little bit, the one about glycine receptors in the spinal cord. I couldn't quite wrap my head around how it related back to taste.
That one might seem like a bit of a leap, but it highlights some fundamental principles of neurotransmission that are actually relevant to how taste signals are transmitted from your tongue to your brain.
Neurotransmission. That's how those nerve cells talk to each other, right?
Exactly. And this particular source focuses on glycine, which is an inhibitory neurotransmitter. It plays a crucial role in regulating muscle activity in your spinal cord.
So it's involved in things like movement and reflexes.
Precisely. Now, the key takeaway here is that glycine, just like those taste molecules we've been talking about, binds to receptors on those nerve cells. This binding triggers a cascade of events that ultimately lead to a change in that cell's electrical activity.
And that change in electrical activity is what allows the signals to travel throughout the nervous system.
Exactly. So even though glycine receptors and taste receptors are located in completely different parts of the body and are involved in very different processes, they both use this fundamental mechanism of ligand binding to generate those electrical signals that can then be transmitted and interpreted by the nervous system.
So it's like learning about the language of those nerve cells in one context can help us understand how that same language is used in another context, even in something as seemingly unrelated as taste perception.
That's a great way to put it. It's about recognizing those underlying similarities in how our bodies process information, even across totally different sensory systems.
I love how these seemingly unrelated sources actually broadened our understanding of those fundamental principles of biology. It's like we took this scenic detour and ended up with this much richer, more holistic view of the whole landscape.
It's a testament to the interconnectedness of those biological systems, and it underscores the importance of exploring these diverse areas of research to gain a more complete understanding of how our bodies work.
This deep dive has been an incredible journey. We explored those molecular intricacies of taste perception, uncovered that whole hidden world of microdomains and AK paths, and even ventured into those realms of thyroid hormone receptors and glycine receptors. It's been quite a ride.
It has. And through it all, we've gained a much deeper appreciation for the complexity, the elegance, and that incredible interconnectedness of our sense of taste.
I'm walking away from this conversation with a newfound sense of wonder, and definitely a thirst for even further exploration. But for now, I think it's time to savor all this newfound knowledge, and maybe even indulge in a delicious treat or two, armed with this deeper understanding of those incredible processes happening on my tongue and in my brain.
I couldn't agree more. Until our next deep dive, happy tasting!